Okay, next slide. Thanks, Wei. Okay, well, good morning, everyone. Welcome to the Coffee Seminar Series. I'd like to introduce myself. My name is Tim O'Hare. I'm a Principal Research Fellow with the Centre of Nutrition and Food Sciences, which is one of the centres within Coffee. Next slide. Thanks, Wei. Just a brief acknowledgement of country. UQ, we acknowledge the traditional owners and the custodianship of the lands in which we meet. We pay our respects to their ancestors and their descendants who continue cultural and spiritual connections to country. Next slide. Okay, just a little bit of housekeeping, everyone. Today's seminar is scheduled to run from 12 to 1. So Wei's talk today will run till about 12:45, after which we'll have a question and answer time. So for you to put in a question, if you can use the tab at the bottom of your screen, the question and answer tab, enter your question, then we'll ask that at the end of the seminar. Please don't use the chat function. We can ask. So remember, use the Q&A function, not the chat function, because we'll miss it otherwise. Okay, next. All right, well, so Wei, we're lucky to have Wei here today because Wei was stuck in China very early on in the COVID pandemic. When people couldn't travel, and he was actually caught in China for about 1.25 years during the middle of his PhD. So we managed to get Wei back about in the middle of last year, so which is terrific. So Wei's, as I said, he's doing his PhD on the fatty acid profile of macadamia nuts with a focus on the really interesting omega-7 oil, palmitoleic acid, which is quite rare. His thesis revolves around identifying the effects of genetic factors, both from the mother tree, but also from the pollenizer or the paternal source of pollen, on the accumulation of fatty acids, as well identifying the candidate genes for fatty acid manipulation in macadamia kernels. So I'd like to pass you over to Wei for his seminar.
So hello everyone. I too acknowledge the traditional owners and their custodianship of the lands on which we meet today and pay my respect to their ancestors and their descendants. And today I will be talking about genetic factors affecting the accumulation of palmitoleic acid in macadamia nut kernels. So macadamia nuts are nuts that are really high in oil, and in fact, 70% of their total weight is oil. And out of those oils, palmitoleic acid is an uncommon omega-7 monounsaturated fatty acid that is present in macadamia nuts in relatively high amounts, usually ranging from 12% to 30%. So the chemical structure of palmitoleic acid is listed down here, and it can be seen that the first double bond occurs from the 7th carbon atom, starting from this side. So this is why it is called omega-7 unsaturated fatty acid. So palmitoleic acid is known to exhibit beneficial biological functions, including supporting cardiac growth, regulating endothelium function, and proliferating pancreatic beta cells. It's also reported to be associated with health and disease, including obesity, diabetes, and cardiovascular disease. So there are two types of fats, saturated fat and unsaturated fat. So saturated fats are fats that do not have a double bond in them, and unsaturated fats are fats that have at least one double bond in them. And it is generally accepted that too much saturated fat is detrimental to human health, and compared with other nuts, macadamia have a relatively higher saturated fat content, usually ranging from 14 to 15%. So it is also widely accepted that the main health benefits come from replacing saturated fat with unsaturated fats, not just from reducing saturated fat itself. So this leads to an interesting hypothesis. So since palmitic acid, C16-0, is the main saturated fat present in macadamia nuts, and it is also responsible for the synthesis of other saturated fats, as listed down here. By increasing the palmitoleic acid content in macadamia, it is possible to reduce the saturated fat and lead to food products with increased potential nutritional and commercial value. So due to its potential health and nutritional importance of palmitoleic acid, there's a need to identify the genes associated with the accumulation of palmitoleic acid in macadamia nut kernels. So a few previous studies have attempted to identify the C16-0 desaturase gene in macadamia. However, these studies were only able to isolate the C18-0 desaturase gene. And this could be attributed to a few possibilities, including one, the desaturation of palmitic acid to palmitoleic acid is catalyzed by a divergent desaturase that belongs to a totally different new family. Second, the C18-0 desaturase is responsible for the palmitic acid desaturation, and its efficiency is dependent on single amino acid substitutions between varieties. Finally, instead of a single gene and enzyme, the accumulation of palmitoleic acid in macadamia is a multi-genetic trait that is affected by various factors. So therefore, traditional cloning strategies may not be useful in this case. Genome-wide association studies, also known as GWAS, has been emerging as a powerful tool to discover relevant genes that are associated with phenotypic traits of interest and have been carried out in oil seeds in recent years. It helped mine alleles for crop improvement and help researchers gain a better understanding of the relevant biosynthetic pathways of fatty acids. So therefore, GWAS may serve as a viable strategy to unravel the underlying mechanisms to how macadamia nuts accumulate high levels of palmitoleic acid. So in order to understand how macadamia nuts accumulate such high levels of palmitoleic acid, there's a need to understand the fatty acid diversity and interrelationships in the current available macadamia germplasm. So this not only helps us gain a better understanding of the fatty acid profile in macadamia, which has not yet been comprehensively studied, but also provides us with the essential phenotype population that is required for future GWAS studies. So in this study, 197 varieties of macadamia across 3 species, including Integralifolia, Tetraphylla, and Ternifolia, have been collected, and including commercial cultivars, elite breeding lines, and wild accessions. And then the oil from 3 individual nuts from each variety were extracted and has been subjected to fatty acid profiling using GCMS. So results from an early scoping study done by my supervisor, Tim, has provided early indications of a wide genetic range for both saturated fat and palmitoleic acid. So it can be seen that the palmitoleic acid initially ranged from 9.6 to 24.5% here, and the saturated fat content ranged from 12% to 17.1%. So the further analysis of these 197 varieties expanded this range, and it can be seen that the palmitoleic acid ranged from 7.8% to 33%, and the saturated fat content ranged from 10.8% to 22.8%. It should be noted that this wide range is extremely suitable for a GWAS study, and this analysis actually expanded the species from only Macadamia Integralifolia to 3 species, and also expanded the cultivars from commercial cultivars only to commercial cultivars, elite breeding lines, and wild germplasms as well. This slide illustrates the top 3 candidates for high palmitoleic acid and low saturated fat content from commercial cultivars, elite breeding lines, and wild germplasm. It can be seen that the highest palmitoleic acid candidate was wild germplasm number 194, ranging as high as 33%, and the lowest saturated fat candidate was elite breeding line number 138, being as low as 11%. And these two may be really useful in future plant breeding. Another important aspect of this study was the identification of important fatty acid correlations. So principal component analysis revealed that the saturated fat showed no correlation with palmitoleic acid, C16-1, meaning that a kernel with a low saturated fat content may either have high or low palmitoleic acid content and vice versa. Another important correlation was that C16-0 and C16-1 are negatively correlated with C18-1, meaning that a kernel with a high palmitoleic acid content is very likely to have a very low C18-1, which is oleic acid content. So this implies that competition exists between these two fatty acids, C16-1 and C18-1. And this is actually the most important fatty acid correlation identified in the study. And more in detail will be discussed in further sections of this presentation. So after understanding the fatty acid profile interrelationships, it is necessary to identify the effects of the genetic factors, including both the mother tree and pollen donor, as well as maturity on the fatty acid profile. So this ensures that correct sampling and experimental procedure could be carried out in this project. And it would also provide valuable information for future plant breeding and also assist in producing food products with consistent fatty acid values. So in terms of the genetic factors, since the kernel is the progeny of both the mother tree and pollen donor, it is important to understand how the maternal genes, and especially how does the father's genetics influence the kernel's oil profile. In terms of maturity, how does the oil profile change with increasing maturity and nut development? So in order to understand the effects of both the genotype maternal parent and pollen parent, a 3x3 pollination trial was designed, including 3 maternal parents BIM that were individually treated with 3 different pollen sources, 741, 344, and 246, and this yields 9 different crosses. And to identify the effects of the maternal parent, the control crosses with the same female parent were grouped together, and the fatty acid profile on 3 different maternal parents, B, I, and M, were examined. So according to this figure, it could be seen that the ANOVA and LSD tests revealed significant differences on all 6 principal fatty acids, including C16-0, C16-1, C18-0, C18-1, C18-2, and total saturated fat content. So as for the effects of the paternal parent pollinizer, the fatty acid profile of controlled crosses were examined, including with the ones having the maternal parent BMI and the pollen donor 741, 344, and 246, the 9 controlled crosses. So the ANOVA tests and LD tests again reveal significant differences in all 6 principal fatty acids as well. And this implies that pollen significantly affects the fatty acid concentration in macadamia nut kernels. So in order to further confirm the effect of the pollinizer, the fatty acid values in both open pollinated and self-pollinated cultivars were examined. And according to this table, it can be seen that in a single cultivar, the standard error in open pollinated kernels were higher than self-pollinated kernels. And this is especially in terms of C16-1 and C18-1, as can be seen here. And again, the ANOVA and LSD results detected significant differences between these. And above all, it can be concluded that pollen actually plays a significant role in oil accumulation and determining the final fatty acid concentration in macadamia nuts. So in terms of the effect of maturity, premature nuts and mature nuts from 3 different cultivars, P, R, and 791 were examined. So it can be seen that during maturing, palmitoleic acid and the total saturated fat content tend to decrease, as can be seen in these 2 figures, as there's a trend here. And the C18-1 content tend to increase, as explained in the right figure, as there's a trend here. And this actually could be partially explained by the general biosynthetic pathway in higher plants. So This is the proposed biosynthetic pathway of the principal fatty acids in macadamia nuts. So it can be seen that before reaching full maturity, the elongation pathway from C16-0 to C18-0 is very weak. So most of the C16-0, which is synthesized from other short chain fatty acids, gets desaturated to C16-1. And it should be noted that there will be desaturation if there should be differences in the desaturation efficiencies between different species and is less efficient than the C16-0 desaturation. So when the nut kernel reaches full maturity, the elongation pathway from C16-0 to C18-0 gets fully upregulated. So most of the synthesized C16-0 gets converted to C18-0 here, where it is then catalyzed by very efficient desaturase to C18-1 here. So varieties may actually vary at what maturity stage elongation occurs, and this would play a huge role in the principal fatty acid content in the final kernel. So if it occurs at a later stage, it will result in a higher palmitoleic acid content. If it occurs at an earlier stage, it will result in a lower palmitoleic acid content. And this actually explains the correlations in the principal component analysis, especially the inverse correlation between C16-1 and C18-1, as well as the changes in fatty acid profile during maturity and nut development. So it is widely known that in GWAS studies, inaccurate phenotype and genotype associations may lead to inaccurate and false positive results. And because both the mother and father play a genetic role in determining the oil profile, it is actually very important in this case to genotype the kernel itself, rather than just the mother tree. And this is especially important when the paternity of the kernel is unknown in uncontrolled pollination situations. So therefore, an efficient DNA extraction protocol from nut kernels directly needs to be developed for the study. So this flow chart here outlines the main procedures of this developed protocol, where nut samples were freeze-dried, followed by buffer extraction, solvent extraction, Chelix 100 extraction, RNA removal, DNA incubation, and finally DNA quality evaluation. So the most interesting procedure in this protocol is the inclusion of Chelix 100. So Chelix 100 is a chelating resin that is used in the extraction of DNA from forensic materials, especially when the DNA is in very low amounts. So it is capable of increasing the corresponding PCR signal of the extracted DNA. However, in our study, we found that Chelix 100 was capable of increasing both the quality and quantity of the DNA, probably due to its ability to remove impurities and contaminants during the extraction process. So this slide shows the quality verification of the DNA extracted using this study. So it can be seen that according to the results from nanodrop, gel electrophoresis, and PCR amplification with macadamia-specific SSRs, it could be concluded that the DNA extracted is of high quality and could be very suitable for PCR amplification and DNA sequencing in future studies. It should be noted that this is the first protocol ever to extract high-quality DNA from macadamia kernels itself. So after collecting all the essential information, as well as developing an efficient DNA extraction protocol from kernels, a genome-wide association study was finally conducted on macadamia nuts. So 168 nut kernels with a palmitoleic acid ranging from 7.8 to 33% that consisted of a normal distributed population were selected using a Shapiro-Wilk normality tests. And the DNA of these corresponding kernels were extracted and sent out for dark sequencing. So in order to increase the accuracy, mainly the imputation accuracy, samples with a minimum of 50% of DNA sequence present and SNPs with a call rate higher than 80% were selected, resulting in a final of 155 samples and 4,787 markers. And finally, GWAS was then performed with LDK and NI imputation, the most conventional method, and using mixed model analysis. So this slide illustrates the QQ plot and the Manhattan plot of the results of this genome-wide association study. So 10 significant markers associated with the palmitoleic acid content were identified, and the false discovery rate was adjusted to the LD threshold of 4.0. And these markers explain up to 24 to 26 of the genetic variants of C16.1 palmitoleic acid. And this indicates that these markers may be linked to genes that play a significant role in the biosynthesis and accumulation of palmitoleic acid, indicating significance. So out of the 10 markers identified in this study, 6 were linked to uncharacterized unknown genes, which may be novel genes, and 4 of them were annotated. And out of these 4 annotated genes, 3 of these candidate genes may be linked to fatty acid synthesis. So for the first one, MA00194, is linked to a gene that is responsible for the expression of a band 3 binding protein and is involved in post-Golgi vesicle-mediated transport. So the Golgi apparatus is responsible for the packaging and processing of lipid and protein molecules, and post-Golgi vesicle-mediated transport plays a huge role in the lipid metabolism and homeostasis in both plant and animal cells. Another interesting finding was that in the previous GWAS study, aiming to find genes related to the oil accumulation and oil seed rape, 3 candidate genes linked to glycosyltransferases were identified. And glycotransferases are located on the Golgi apparatus. So since this is a significant marker identified in our study, it may be linked to one of those glycosyltransferases or products. As for the second one, MA00614, is responsible for the transport of an oxidoreductase A into the chloroplast. So this enzyme is a reductase that catalyzes a very active light-dependent reaction in the chloroplast and uses up a lot of NADPH. It is widely known that the NADPH in the chloroplast is essential for fatty acid biosynthesis, especially during the elongation step. So competition may occur between these 2 processes, and this may explain why this is a significant marker identified. And finally, the last one, MA02000, it is linked to a gene that is responsible for the expression of a RAN binding protein and is involved in nuclear regulatory protein transport. So previous studies have provided substantial evidence that a wide range of regulatory proteins in the nucleus are responsible for the expression and regulation of fatty acid synthesis genes. And there is a possibility that this marker is linked to one of those regulatory proteins of interest. So one interesting finding in the study is that we did not identify genes directly linked to known desaturases and elongases in macadamia nuts. And this could be attributed to a few possibilities. So first, the annotation of the reference genome of macadamia is not complete yet. And it can be seen that a huge number of unknown genes that were uncharacterized were discovered in previous slides. However, this is the only reference genome in macadamia that is available, and this is the best that we can do currently. And second, to increase the imputation accuracy, only SNPs with a call rate higher than 80% were used. So only 4,700 markers out of 9,800 markers were used. And actually half of the markers available were not included in this study. So those that were linked to desaturases and elongases could simply been left out and not included in the final analysis. And finally, instead of being controlled by genes in a single regulation pathway, there are novel genes responsible for the accumulation of C16.1 palmitoleic acid. And this is actually supported by both published and unpublished material. So previous studies did not identify any novel desaturases that were responsible for the biosynthesis of C16.1. And we also attempted to find these novel desaturases using primers, both degenerate ones and primers designed based on conserved regions of known fatty acid desaturases in macadamia. However, only the C18-0 desaturase was identified as well. So in addition, we also try to sequence the C18-0 desaturase in both high and low palmitoleic accessions in macadamia nuts. However, we did not find any SNPs that could have been responsible for the difference in palmitoleic acid content in these accessions. So above all, these results may suggest that if a C16-0 novel desaturase did not exist, and the C18-0 desaturase is actually responsible for both the synthesis of C18-1 and C16-1, the C18-0 desaturase may actually not be a key factor in the accumulation of palmitoleic acid. So in recent years, previous studies has also provided evidence that other than SNPs, regulatory factors that control the expression rates and levels of fatty acid synthesis genes also play a huge role in oil accumulation in higher plants. But it should be noted that a lot of these regulatory elements are actually outside of the fatty acid synthesis pathway, such as hormones and regulatory proteins. So the genes identified in the previous slides may actually be novel genes that are responsible for these regulatory pathways. So next, we also conducted a genome-wide association study using the conventional method with DNA extracted from maternal young leaves rather than from kernels. So instead of using individual fatty acid values from one kernel, the phenotypic data in this study consisted of mean palmitoleic acid values from 5 individual kernels of a single macadamia cultivar. And as explained before, the DNA was extracted from maternal young leaves. And in a total, 122 macadamia cultivars and 8,875 markers were included in this final study. So by doing both of these methods, it is possible to compare the advantages and disadvantages of both methods. So in this study, only 2 significant markers were identified, including one linked to an uncharacterized unknown gene and one linked to an annotated gene. However, the function is a bit unknown as it is currently unrelated to fatty acid biosynthesis. Interestingly, these markers only explain up to 2.54 of the genetic variants, indicating that these markers may not have played a huge role in the accumulation of palmitoleic acid in the final nut kernel. So taken all together, these results suggest that the pollen needs to be taken into account. And using kernels as the source of genomic material for GWAS in this case may actually be more accurate. And this slide concludes the advantages and disadvantages of using both methods. So in terms of the kernel method, one important advantage is that it's fairly easy to get a normally distributed phenotypic population. All you need to do is just phenotype a population and handpick out those that consist of a normal distribution curve. The most important advantage is that it provides reliable genomic material as it takes account of pollen, including both maternal genes and paternal genes. However, it should be acknowledged that the quality of DNA extracted from the kernel is inevitably poorer than those extracted from young leaves. So this results in missing data in the sequence, and as a result, less cultivars and markers could be available for the final study. As for the maternal method, the main advantage is that good quality DNA could be easily obtained, resulting in less missing data and more cultivars and markers included in the final study. However, using the maternal method could possibly lead to inaccurate phenotype and genotype associations, as it does not take account of pollen and is very difficult for the final population to be normally distributed. So finally, this reaches the conclusion of this presentation. So first of all, fatty acid biodiversity exists in the current available macadamia germplasm, and there are important interrelationships, especially the inverse correlation between C16-1 palmitoleic acid and C18-1 oleic acid, indicating competition. Second, the fatty acid profile of macadamia nut kernels is influenced by both the seed parent and pollenizer. And due to the effect of pollen, it is necessary to use the nut kernel as the source of genomic material for GWAS on fatty acids in macadamia. Third, the fatty acid profile in macadamia is affected by kernel maturity and upregulation of the C16-0 elongase during kernel development possibly plays a crucial role in determining the concentration of the principal fatty acids in macadamia. So fourth, and this is possibly the most important conclusion, is that this is the first GWAS ever to be conducted on macadamia, and the first GWAS study ever on any plants to use kernels as the source of genomic material and to compare the advantages and disadvantages of both methods. And finally, this study may have identified a couple novel genes associated with the accumulation of palmitoleic acid in nut kernels of macadamia. And the exact function of these genes need to be verified in the future. So thank you everyone for listening to my presentation.
Yeah, thank you, Wei. That was excellent, an excellent presentation. Just to remind everyone, if you've got questions that you're thinking of during this question session, write them in the Q&A and we'll endeavour to ask them. Okay. Now, Wei, first question from Craig Hardner. Could variation in the oil profile among varieties be explained by variation in efficiency itself rather than indirectly through variation in maturity?
So does the variation in efficiency itself mean the efficiency of enzymes involved in the biosynthetic pathway?
Yeah, like efficiency in the desaturase enzyme.
Yeah, that's actually very possible. But in one of the unpublished studies done by us, we actually sequenced excuse me, we actually sequenced the C18-0 desaturase in both high and low palmitoleic acid accessions. And that is actually the only desaturase that has been known in macadamia currently. We did not find any SNPs that could have been possible, that could have been responsible for the difference in palmitoleic acid content, but that's definitely possible. It could be actually influenced by various number of factors, including SNPs in the efficiency, SNPs that affect the maturity, as well as like other regulatory proteins that affect the overall expression level of these fatty acids.
In fact, Craig's just remarked maybe his first question's redundant. Now I think you answered it during the thing. So, but yeah, that, I mean, it was interesting that there was no desaturase that specifically came up in the genome-wide association study in particular, which we. I have another question from Craig. There's a slightly different area. You mentioned that the oil content of macadamia is one of the highest across different nuts. Do you have any thoughts on the biological reason for macadamia to produce seeds with very high oil content?
It could be based on other plants. It could be that has a really so the rate limiting enzyme that in the fatty acid biosynthesis step, at the first step, that uses acetyl coenzyme A and goes into malonyl coenzyme A. So it could have a really efficient enzyme of that, which actually contributes to the whole fatty acid synthesis rather than one. But this is just a possibility. So
from an evolutionary point of view, though, why would I think it means why would the nuts have such a high oil content? And I can make a suggestion if you like. I know,
yeah, sure.
Craig, in published literature on the proteaceae, which macadamia is one of, you get a wide range of oil contents. You find that rainforest plants, such as macadamia, tend to have high oil or energy sources. And the theory is to enable them, because they're growing in shade, they need to grow up and reach the light, as opposed to something like a grevillea seed growing in a more heathland situation where there's lots of light, but the soil's very sandy, and it's more important for those to have high stores of minerals by comparison, and oil is not important. So you might have a much drier seed. So that was a theory brought out by the West Australians a few years back about that. Okay. Okay, another question, Wei. So considering the kernel is an embryo from a maternal tree and a pollen donor in macadamia, does that mean that most oil profiles published previously are really reliant on whatever pollinizer just happened to be present in the field at the time? And so I suppose how accurate are these oil profiles that have been published, but maybe both for macadamias and for other tree nuts?
Oh, okay. So for the pollen effect, also known as the Zenny effect, and the fatty acids. So actually, it is actually very dependent on the plant species, and it has actually been proven that in maize, soybean, and like certain oil seeds, pollen does actually play a role in determining the oil accumulation and in the final kernel. And as for macadamia, it seems to be especially important, especially according to the results of our study. It can be seen that the LSD results, pollen actually did play a really significant role in determining the final oil concentration and fatty acids in the controlled crosses. So I would say that it is actually very dependent on the pollinizer, and if you don't take the account of the pollinizer, it might not be very accurate as a whole. Yeah.
Yeah. So because I know that a lot of food scientists go to the local supermarket and buy a packet of macadamias or other tree nuts and whatever vegetable, but specifically in tree nuts, which are an embryo and have this paternal and maternal effect. They these results, they measure them, they get a result, gets published, but it, like macadamias, quite often because there's such strong outcrosses, that at least two cultivars are grown in the field, sometimes in alternate rows, and to increase nut set. So depending on what that second cultivar is, or the pair of cultivars, I imagine would definitely have a strong effect on the final fatty acid profile and palmitoleic acid in the macadamia nut.
Yeah, that's absolutely. It will definitely affect it significantly.
Okay, another question. Do you think there is actually a C16 desaturase, or do you think it's just a C18 desaturase acting on palmitic acid to convert it to palmitoleic acid?
Well, based on previous studies, no novel desaturase that specifically catalyzes C16-0 has been discovered yet. And in previously published paper, finding the relevant C16-0 desaturase in cat's claw, they also did not find any divergent ones, but found one that was really close, really similar to a C18-0 desaturase, but only had a few amino acid substitutions in the substrate binding pocket. So it could be a couple possibilities. A divergent one could exist, but I think the possibility is low because having an enzyme a fatty acid enzyme that's completely in a totally new family and a totally new sequence seems like a rare occurrence to me, because most fatty acid desaturases have a conserved region that in different species. It could be that the C18-0 desaturase also catalyzes the desaturation of palmitic acid, but maybe it may have amino acid substitutions differences and affects the efficiency as well. So you
think
that's also likely a point. Yeah.
Okay. So you think the maturity hypothesis about the stage at which different accessions upregulate their elongation from the C16s to the C18s could be a key mode of controlling how much palmitoleic acid?
Yeah, absolutely. The elongase could be a really important factor in determining. And actually, in something that I didn't show here today, we did find one candidate gene that is related to flowering and maturing of the plant that's associated with palmitic acid, which is C16-0, and that could actually mean something because it affects the maturity and somehow affects the upregulation of the elongase as well.
Okay, now that'll be really interesting. I have one more question. I noticed in your genes that you identified that one of them mentioned chloroplasts, and that macadamia kernels don't have chloroplasts. Can you comment on that?
Oh, okay. So the reference genome is likely to be annotated on maternal leaf tissues rather than kernel. And so when comparing these functions, there might be a bit of a difference because there are different organelles in plant, like leaf cells and kernel cells. However, because the sequence is very similar, it's almost identical, I would assume that it has a similar, very similar function in the kernels. So in the kernels, fatty acid synthesis occurs in the plastid rather than chloroplast. So I'll assume it's an enzyme that catalyzes the same reaction and uses up NADPH in the plastid rather than the chloroplast. So yeah.
Yeah, okay. Yeah, no, I'd agree with that, that plastids, there's a very close relationship between chromoplasts, chloroplasts, amyloplasts, and other plastids, such as those generating the fatty acids. So yeah, it could well do that. Okay, well, that's the last of our questions for today. Thank you very much, Wei. Okay. Just like to give a little bit of advertising for the Coffee Science Seminar page. If you're interested in this and other seminars, please go to the Coffee Science page and you can scan it there. You can give feedback on seminars if you're keen. And oh, hang on. Can you go back a slide, please? I think we've gone forward a slide, Wei. Okay, there we go. And just advertising the next seminar. The next seminar, not almost as exciting as fatty acids and macadamias, as sex and drugs. So by Dr. Alistair McTaggart. If you can clock in next week, same time, same channel, you can have a good show, and I'm sure it'll be very exciting by Alistair on his take of sex and drugs. So next slide. And we've done that one. Next slide. And that is it, everybody. So thank you very much for attending.
Oh, thanks everyone for coming.